On.
Oh, it's a the arrow on the video.
Great name on it. You increase your confidence in rejection.
The apple doesn't work. It's weird. Oh, well. It's I guess turn the video off. Oop, turn the video off.
Name is like Smith.
Maybe you can middle initial, then you can eat our middle name billboard, maybe. I don't think he ever publishes with it though, does he? I think he's always, it's just.
No, I never stop you because it's on his AAR.
Amazing. There are a few, does it?
All right, everyone. Hi, good morning and good afternoon, and welcome to the final quantitative seminar of the spring quarter. Congratulations, everyone, for finishing up research, finishing exams, finishing proposals, all that. We've made it to the end of the year. Give ourselves a mini round of applause. Good work, everyone. And welcome. I'd like to first begin with the land acknowledgements. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. And I'd like to introduce today's speaker, Kathleen. Kathleen is in her third year pursuing a PhD as part of the Roberts Lab in the UW School of Aquatic and Fishery Sciences, SAFS, studying epigenetics and the mechanistic basics of stress response in important marine invertebrates like corals and oysters. Please give me help me giving a warm welcome for Kathleen.
Thank you. Hi, so I'm Kathleen. I'm going to tell you about some portions of my thesis work looking at epigenetic mechanisms in corals. So first, I'm going to show you all have a good grasp of what epigenetics is. So all of us inside our cells have DNA, which essentially forms the instruction manual for your body. It is information on how to produce all of the necessary components for cellular function. And epigenetic mechanisms are mechanisms which influence how that DNA is being used. So when genes are being turned on or off, things like this, without actually altering the DNA itself. So you can think of them, if DNA is like your body's instruction manual, these epigenetic mechanisms are just annotations that are sitting on top. So they influence how the manual is being read, but not the words that it actually contains. We are going to have to get a little bit more into the weeds, though, before we can talk about my research. So this is the sort of standard flow of information, the central dogma from DNA to physiology. So DNA is processed into messenger RNAs, which are then used to create proteins, the action molecules of the cell, and these ultimately determine your physiology, your phenotype. But because we have this epigenetic component, this flow of information is incomplete. There are a whole range of different epigenetic mechanisms which can influence physiology, just sit on top of DNA and influence physiology. I'm just going to talk about three that are the focus of my research. So the first is DNA methylation. This is one of the best studied epigenetic mechanisms, and they're essentially consists of chemical tags, methyl groups that are fixed to the DNA outside of the nucleotides, and it influences how that DNA is being read. In vertebrates, DNA methylation is pretty well understood. Basically, the addition of methyl groups to the gene promoter turns the gene off. In invertebrates, it doesn't work that way. It's distributed differently across the genome, and we don't really know mechanistically what it does yet. We just know that it is doing something. It's associated with physiological change and is inflammatory responsive, things like that. Next, I look at something called microRNAs. These are a class of non-coding RNAs, so RNAs that do not encode for a protein but still have cellular function. And microRNAs are, as the name suggests, a very, very small class, roughly 22 nucleotides in length. And they function by targeting complementary portions of messenger RNA molecules and then recruiting protein machinery to either inhibit translation or break down the messenger RNA completely. So they're silencing mechanism. And then finally, long non-coding RNAs. This is another class of non-coding RNA, but they're much larger and more diverse than microRNAs. So long non-coding RNAs are any non-coding RNAs over 200 nucleotides in length, and they can act at the DNA, RNA, or protein level, and they can do a bunch of different things. They can act as guides or decoys for other molecules. They can help scaffold enzyme concept complexes. They can do a lot of different things. So similarly to DNA methylation, they're difficult to study, particularly in non-model organisms, because it's hard to understand what they're doing. So these are the three I focus on in my research. Importantly, all three of these mechanisms are environmentally responsive, associated with physiological change, and reversible. This is why they're so relevant for ecological work, because they essentially facilitate response, so environmental response and acclimatization. Also, I'm going to give you a little sneak peek notification that not only do these layers interact or have effects individually, but they also interact with each other. And so we're going to get into that a little bit later, but it adds an additional layer of complexity to the epigenomic landscape. So now that we have sort of done a crash course on these epigenetic mechanisms, I also want to make sure you understand just how cool epigenetics is and how impactful it is across scales. So I'm going to give you a quick example outside of the marine invertebrate world. These are locusts. So locusts are swarming insects that form these huge aggregations and sort of swarm across the landscape and just devour everything in their path. So understanding locust behavior and what causes locust swarms and things like that is relevant because they have huge ecological, agricultural, economic consequences. And actually, locusts are just a phenotype of grasshoppers. So some species of grasshoppers, whenever they experience high population density, there's a density-dependent phenotype shift. So high population density causes a shift from what's called the solitary phenotype to the swarming or gregarious phenotype, which is what we consider a locust. So on the right, these are both in the same species at different life stages, but on the right is the solitary phenotype, or left is the solitary phenotype, and on the right is the swarming phenotype. And it turns out one of the important mediators for this phenotypic shift is an epigenetic mechanism. It's a microRNA. So there are actually two, but this study looks at one of those microRNAs, miR85P. Whenever the grasshopper experiences sort of signals of high population density, so it can be like, for example, rubbing on their back legs because they're just pressed up against so many other grasshoppers. Whenever they experience this cue, miR85P is upregulated and inhibits specific neurological pathways, which results in a phenotypic shift from the solitary phenotype to the gregarious phenotype. And then importantly, this change is also reversible within the individual's lifetime. So after they're done, once the trigger is gone, microRNA is deregulated, and then they can transition back to the solitary phase. So not only is it super cool to understand this mechanism, it's also very useful to understand the sort of epigenetic bases of these phenotypes. So for example, in this paper, they were able to develop a transgenic rice line that expressed an inhibitor of this microRNA, so that then whenever the locusts fed on this transgenic rice, it reduced the levels of the microRNA in those locusts, which resulted in a de-repression of the trigger pathways and shift back into the solitary, non-swarming phenotype. So basically, epigenetic manipulation is a potential avenue for locust control, something other than pesticides, which could be really useful. So, epigenetics, they're complicated, but they're really cool, and they can have a wide range of effects. And it's important to understand them for these reasons. So now we're going to move into corals, which is the study system I work in. So, as we've established, epigenetic features provide dynamic, reversible mediation between environment and phenotype. And this is particularly relevant for corals who live in really dynamic environments and are undergoing environmental shifts, as are most organisms due to climate change. The corals are sessile colonial marine vertebrates, animals. They're sessile, which means they are stationary, they're fixed to a substrate in which they live, And colonial means that actually the whole coral organism consists of many, many, many little coral polyps. The polyps, the coral organism as a whole, has a mutualistic symbiotic relationship with photosynthetic algae. So if you see inside this translucent polyp, all those little green brown dots are the algae that live inside the coral tissue. And these algae provide a significant portion of the coral's energy budget, so it's an obligate symbiosis. Corals, stony coral rather, build these calcium carbonate skeletal structures that form the foundations of reef ecosystems. The reef ecosystems are, of course, very important. This supports 25% marine biodiversity worldwide. They provide coastal protection, sustain fisheries, and have cultural, economic, and ecological relevance. However, they're also quite vulnerable, and the climate crisis is exacerbating that. So, as I mentioned, corals have this obligate symbiosis with photosynthetic algae, but stress to the coral can cause a breakdown of that relationship, which forces the coral host to eject the algae into the water, leaving a bleached coral, which is not a dead coral, but it's one which is much more vulnerable to continued stress. And bleaching does often lead to mortality, usually through starvation. And while there are many types of stressors which can cause bleaching, by far the most prevalent is thermal stress. So this plot just shows the blue line and area bars are global coral coverage. And the vertical red bars represent anomalies in sea surface temperature. So basically marine heat waves. And as marine heat waves have become more intense and frequent, so has global coral coverage decreased. So the climate crisis is a real problem for corals. And the biggest problem is that it's happening so quickly that adaptive evolutionary response can't keep up, which is why it's becoming important to understand other avenues for response and persistence. And this is, again, where epigenetics comes in. Because adaptive responses are really too slow to ensure persistence of corals and coral populations, we need to be looking at methods for fast, dynamic response that can happen within a single life. So, this is where we have a break. Corals do have epigenetic features. There are epigenomes that are functional physiological implications. The problem is we don't know exactly how they work because many of these features mechanistically differ in corals than they do in the more well-studied vertebrate and mammal organisms. So my research is attempting to answer this question of whether we can use these epigenetic features to predict the coral's physiology and ultimately response to environmental shifts. So we now come to my study system. I'm working with samples collected from Moorea, French Polynesia. The study had 2 sites and 3 species of coral covered. So there were tide colonies of Acropora pulchra, Ocelotropora tubulensis, and varieties edemanae. I selected these because they cover the diversity of corals. So they have different growth morphologies, different life histories, different symbiotic associations, different degrees of environmental tolerance, stress tolerance, and they also span the coral phylogenetic range. So if we're going to try to understand how genetics works in corals, we want to really cover our bases, make sure we're covering the full diversity of that genetic variation. And then for each of these 3 species, there are 10 tagged colonies, and these colonies were sampled 4 times over the course of the year. So it's a repeated measure study. So for each colony, we have multiple samples at different time points, and the time points were selected to capture seasonal and biochemical variation. So the extremes of light, temperature, rainfall should all be hopefully encapsulated within our samples. From each collected sample, 4 molecular features were evaluated. First, we did RNA sequencing to measure gene expression, so expression of messenger RNAs, but also of long non-coding RNAs. We also did small RNA sequencing, which captures microRNAs. They're too small to really be captured in traditional RNA sequencing. And then finally, whole genome bisulfite sequencing to map DNA methylation across the genomes of these. So first, we've done just a broad characterization of these epigenomes. And the biggest takeaway is that there's interspecific variation. So while the counts of these features are in some cases pretty similar across species, the biggest takeaway is over here, it's probably too small font, that there's very little conservation among species. So there's only 4 microRNAs which are conserved among all 3 species, a tiny fraction of 46 long non-coding RNAs that are conserved, and then I don't have the count up here, but only a small number of orthologous genes were methylated in all 3 species. So the reason this is important to know is it means it's not necessarily helpful to be looking at individual features. So, for example, it might not be that useful to focus in on a specific microRNA to understand what it's doing, because that microRNA probably doesn't exist in any other corals, or most of the coral species. So instead, we should be focusing on sort of high-level characterization, understanding broadly what are microRNAs doing in these corals, what are long-term RNAs doing, etc. So then we looked at some of the interactions that we can characterize mechanistically. So for microRNAs, we know how they work. They bind to target RNA molecules through a high complementarity region. And so we can look at predicted binding to evaluate what the microRNAs might be targeting in our samples. And we found some interesting instances of these cross-layer interactions that I hinted at. So first, we found a bunch of epimicroRNAs. So these are microRNAs that are targeting the transcripts that encode other epigenetic protein machinery. So for example, microRNA might target the transcript that encodes a DNA methyltransferase, and that's the enzyme that's responsible for maintaining DNA methylation across the genome or assigning new DNA methylation tags. We also found epigenetic microRNAs involved in a whole bunch of other epigenetic pathways. So histone modification, chromatin signaling. This one's fun. We found microRNAs that are targeting components of the microRNA pathway. So components that are involved in genesis of microRNAs or in their silencing function. So not only is there regulation among different epigenetic layers, there's also kind of auto-regulation for these microRNAs. And then on top of that, we also found a bunch of something called competitive endogenous RNAs. So this is essentially long encoding RNAs that also bind to microRNAs and can therefore basically sequester them away from their intended messenger RNA targets. So there's this indirect de-repression from microRNA targets happening with long encoding RNAs. So the reason this is important is, the reason this is important is that not only not only are the layers themselves difficult to characterize in these non-model systems, but there's also an additional added complexity from potential for cross-layer interactions. So not only can we not be looking at individual features because they're locally conserved across species, we maybe shouldn't even be looking at individual layers because the layers themselves can interact with each other. So instead, we shifted our focus to how can we be modeling them jointly to predict outcomes, because the microRNA might not just be regulating physiology, it might be regulating methylation, which regulates. So we need to investigate them in combination to get a sense of the full picture. So now we come to the real quantitative science stuff with our model approaches. There are some challenges to modeling by nature of the data set. The biggest ones are that we have comparatively very few samples for a really high dimension predictor set, because there's so many long encoding RNAs and so many genes that are methylated, just vastly outnumbers the number of samples we have. And then additionally, there's a problem with multi-collinearity because a lot of these features could just be co-expressed or co-regulated. So for example, if a bunch of if a group of long encoding RNAs are involved in regulating the same pathway, they'll probably all be expressed in concert with each other. And so both of these complicate trying to predict expression from these epigenetic predictor sets. Regression can be well suited to handling these like more predictors than sample sizes and multicollinearity. And there's two primary types of regression approaches. There's L1 and L2, or lasso and bridge. Basically, the gist of it is that lasso regression, you can think of it as being a harsh editor. It basically promotes sparsity. So that means even though you have a really high dimension predictor set, it's going to try to reduce the number of relevant predictors that it identifies to the smallest number possible. And so the coefficient of most of your predictors is going to be 0. And this is useful because it means that the output is very interpretable, because you're left with only a small number of relevant predictors that you can then investigate further. The problem is you might be missing things, and it doesn't handle multicollinearity very well. That's where bridge or L2 regression comes in. This one is not sparse. It means every single predictor is retained in the final predictor set. It just might have a very low coefficient or degree of importance for the prediction, but it does handle multicollinearity well because it can retain all these correlated features. Since neither of these are perfect, though, we ended up going with something called the elastic net. So elastic net is, for sure, it's just a combination of the two. So you can make use of the strengths of both L1 and L2 regression, which means that elastic net regression produces sparse outputs, which makes the outputs more interpretable, and it's stable under high multicollinearity. So this is what we're going to get. Basically, the gist of it is we have a predictor set and a response set. The predictor set is the combined set of miRNA expression, non-coding RNA expression, and gene body methylation. And then the response set is gene expression. Elastic net regression is essentially fitting a model for this predictor set on an individual gene. So it's not really computationally practical to try to run it for all of the 20,000 genes in our response set. So instead, we are focusing on selections of genes that are relevant to the questions that we're asking. So for example, we might reduce down to genes that are involved in biomineralization or genes that are involved in energetic state. So we have this predictor set, the epigenetic predictor set, run elastic net to predict the expression of a selected subset of genes with an 80-20 training testing split and bootstrapped amplification. It's implemented through another package called glnet. And then this alpha value 0.5 basically means we've been using an equal split between L1 and L2 regression, but you could alter to change that value to side more with one or the other in your elastic net. And frankly, I just haven't tested different options yet. So I have that check. So this is the high level understanding of how the model training is working. To validate the model, we just use noise injection. So these figures are showing on the x-axis, these are all genes that are involved in the gene set that I was trying to make the response of. And then on the y-axis is R squared. That's basically a measure of model performance. And so if you run a go predictor data, you see that there's a large number of genes that seem to be consistently well predicted by the model over the bootstrapping iterations. But then the question becomes, is that capturing real data, or like real signals, or is it just maybe overfitting, something like that? So to check, we basically just reran the model with shuffled data noise, and just like we would hope to see, the model no longer worked. There was nothing that was consistently well predicted, and that's good. It means that whenever we're running with the real stuff, there is some underlying so there's some sort of genuine predictive structure. Okay. Once we felt confident that the model was capturing real signal, then we need to try to figure out for individual predictors how to assess chromotypal significance. And so first, I tried permutation testing because that's like the standard thing you try whenever hypothesis testing doesn't work. And the idea behind this is that for each predictor, you basically use the response to build a null distribution for that predictor. And then whenever you run it with real data and you get a value, you can see where that value, say 0.045, sits on the null distribution and evaluate significance from that. The problem is that because ElasticNet introduces sparsity, every time we run the model, the vast majority of your roughly 30,000 predictors will have zero coefficients because you've asked for a sparse output. So only something like 50 predictors will have a non-zero coefficient, which means that for any one predictor, whenever you're running this permutation, the null just becomes almost zero. And that means then whenever you run with real data, any non-zero coefficient is assigned significance. But that's obviously not that helpful because that means that whenever I would try running this, every single predictor I got was significant. And that's not what I'm looking for. So instead, we switched to something called stability selection. So stability selection, instead of asking, is this coefficient significant? It asks how consistently is it selected whenever you run the model. So if you basically subsample your observations and refit the model, and then just calculate the probability, the proportion of iterations in which a given predictor was selected as relevant to predicting that gene. And so then I've been using exceptional 0.6, so then if a predictor is selected in 60% of the runs, consider that stably predicted. And so you have more confidence that it's actually relevant to predicting the expression of this gene. Okay. So we're confident that the model is capturing real signal, and we now have a way to better assess confidence in the individual predictors themselves. And so now I can get into some example results. So this is all example of the model being run on, it's just with data from Acropora pulchra, one of our 3 species. And the gene set that was selected for use is genes that are involved in energetic state. And so first, epigenetic context seems to be predicting expression for a good portion of these energetic state genes, which is cool. So over the iteration of the bootstrapping, roughly half were consistently well predicted. And then we can look at, of those, roughly 45, 50 genes, we can look at the composition of their predictors. And we see lots of interesting things. First, all 3 of the predictor types represented, which is nice, including this weird one that was really influenced by microRNA. But generally, long-encoding RNAs are dominating the predictor set. And that's true even if you take into account the fact that there are so many long-encoding RNA predictors, like way more than there are microRNAs. Just proportionally, they're overrepresented in the predictor set, which is cool. And so something I'm really interested in is that I want to do next is rerun this with other gene sets that are on different functions and see if that's consistent. Because one hypothesis I'm interested in testing is whether different molecular layers, preferentially regulate different physiological functions. Anyways, and then you can also dial even further in and look at these individual genes. Like, so this is one that I pulled. It's the one on the left side that has this huge microRNA contribution, and this is cryptochrome 1. That's the gene. This is a circadian clock gene. So in mammals and humans, we think of our circadian rhythm as regulating our day-night cycles. But in corals, it's believed that this gene and others related to circadian clock rhythm are actually more important for reproduction, because many corals reproduce through something called mass spawning, where all of the individuals of the same species at the same time release gametes into the water, and they mix and fertilize and settle. And they have to have some way to synchronize that behavior. And so it's believed that cryptochrome 1 helps synchronize behavior to lunar cycles, actually, which is cool. I'm looking at this see, yes, it seems to be dominated by its microRNA. And then because it's a microRNA, and this is the only feature for which we understand the mechanism of how it works, we can actually do additional structural verification. I found that yes, this microRNA not only is it identified by the elastic net as being important to predicting this gene regulation, it also is predicted to bind to the transcript itself, which is super cool, but also a nice sort of external validation that, again, models capturing those. And you could be doing this type of dialing on any of the other genes that you found interesting or important as well. So, takeaways. First, before we even got to the elastic net, we found that looking through our data, there's a lot of additional complexity we weren't necessarily expecting, and that means we need to be considering the epigenetic integratively. And once we did that, we attempted to do that using an elastic net regression approach, and it seems to be capturing real signal, and it stably predicts the expression of a portion of physiologically relevant genes. Also nicely, the elastic net approach is interpretable because of this sparsity. It means it's feasible for us to be looking at individual predictors of individual genes, if you like, and summarization of things are more feasible. And then perhaps the best part of the study approach is that it doesn't rely on prior information. So you don't need to know necessarily what the gene you're trying to predict the expression of does. That's nice because in non-model organisms, often annotation, like transcriptomic annotation, is pretty incomplete. We don't know what a lot of these squirrel genes do. And we also don't need to know mechanistically how these epigenetic features function to include them as predictors. Again, that's nice because we don't know mechanistically how they work a lot of times, especially for gene methylation and RNAs. There are still some limitations, of course. Obviously, correlation is not causation, so these can help us perhaps narrow scope onto features of interest, but doesn't necessarily prove a mechanistic basis of influence. It's a relatively limited sample size for the number of molecular features, which always complicates things. And then there's also some limitations in how we can compare across species because they have different genome qualities. And that's just something that we haven't gotten in mind before I can make direct comparisons. But for what's next, while all of this is super, super cool, and I find this like mechanistic side of things really interesting, ultimately the questions you're trying to answer are related to physiology. So now that I have a method to, I guess, narrow scope down to epigenetic features of interest, I'm going to be trying to relate the epigenetic state to physiological metrics. So I didn't tell you this whenever I was overviewing our data set, but we also have physiology data, host and symbiote physiology data, and symbiote composition data for all of these samples. So I can be trying to associate these epigenetic features with fluctuations in biomass and calcification, respiration, all those things. And then on top of that, we want to be considering the symbiote because the coral is not just an individual organism, holobiont that is deeply, its physiology and molecular relation is deeply integrated with that of its symbiotic partner. And there's a lot of really interesting work in other symbiosis, especially in plants, that indicates that the epigenomic context plays a huge role in regulating and mediating that symbiosis. And so I'm really excited to see what I can find in corals as well. And ultimately, this should hopefully more information about how physiology and symbiosis is mediated will also help us predict organismal response, persistence under environmental shifts. And I'm happy to take any questions.
We'll start with questions in the room.
I have a very naive question. Is it possible for symbionts or viruses or retroviruses to change the epigenetics of the coral?
Yes. So in corals, this has been sort of lightly investigated at a correlation level in all three of these mechanisms actually individually. So it's mostly been done with anemones and aposymbiotic and symbiotic anemones because you can do that in that system. And symbiotic state is associated with differential methylation patterns, changes in microRNA expression, and changes in monocoding RNA expression. So at least the presence of a symbiont changes how the host epigenome is working. And then on top of that, this hasn't actually. There's also a study in corals where I don't remember which direction. I think it was that symbiont microRNA was found to have targets in the coral host transcriptome. So there's also evidence that the epigenome of the symbiont is regulating things inside the host cell as well.
Thanks. This is also a naive question, probably. Going back to your example with the grasshoppers and how one could kind of like engineer the crops so that they would influence the epigenetics of the bug itself.
Mm.
So bringing that over to the coral example, right? What conceivably do you think that there is scope for kind of engineering corals that are more resistant to this and that condition? Is that something that is already being done? Is it something that could kind of like, is that an avenue for restoration and replacing corals that are kind of dying out in various places?
I think it could definitely be an avenue. To my knowledge, I don't think it's been pursued yet, but definitely. There's a caveat in that, again, at least for the non-coding RNAs, they're very lowly conserved. So if you were going to develop an approach to, I don't know, develop a coral that you change in some way that this microRNA is expressed because this microRNA is associated with increased thermal tolerance. You wouldn't necessarily be able to do the exact same thing to every coral species because that microRNA might not be present in every coral species. So there's conservation limitation. But it's definitely interest, especially the DNA methylation, because it's, I guess, more consistent across species, and it also has the potential to be heritable, so inherited by the next generation.
Another totally naive set of questions. Firstly, I don't think I can see my friends in the Great Barrier Reef wanting to create transgenetic Great Barrier Reef and finding out what happens afterwards. But then,
I think the focus is influencing symbiotic composition right now. Thermally tolerant.
A couple of thoughts. One was, given the way you presented your results, in the sense of types of impacts, whether the lasso regression would come up with qualitatively similar results. In other words, the micros versus the whatever the others were. So because there's clearly going to be correlation in functionality amongst the predictors. But it'd be interesting to see if you just did a straight lasso regression, how that would come out qualitatively.
Yeah.
So one
about changing the next words.
Yeah, the other thing, and this is super naive, I wasn't sure what you're predicting. So when you talk about gene expression, I'm naively thinking you either express or you don't express, so it's a binomial output. But I presume it's not, because otherwise you wouldn't have done what you did. So what are when you're doing these regression, what is the what are you predicting, and how do you express the predictors? Maybe I'm the only person who needs to know that.
So it's transcript counts. So the gene, as soon as the gene is or is not expressed, it might be highly or lowly expressed. So during one time point, it might have 2,000. You have 2,000 transcripts of this gene, and then another time point you might have 50. And so
it's an
it's a
integer or proportion?
It's a count. It's an integer.
It's an integer.
Well, and then before everything was fed into the model, everything was z-score scales, because all of these three different features are operating on different scales. So for example, microRNAs tend to be more lowly expressed across the board to fauna, including RNAs. So the individual layers are counts, but then when they were combined into a single predictor set, everything was Z-score scaled within later.
You don't think you need to deal with the fact that the data really are integers? Well, they're counts, right? So normally we don't, I mean, R squared sort of means something for count data, but normally we would use a count-based regression approach, not a traditional lasso regression, which is really set up for real numbers. Now, if the numbers are huge, that doesn't matter. But if you've got 3, that I be a little skeptical about how the whole sort of regression builds itself. So is that just not done in the field, or?
I'm not sure what the question is.
Well, these aren't real numbers, right? They're integers. Even if you Z-score them, they're going to be discrete. Is that not something of concern to anybody? So if we're doing a normal regression of count, you use a likelihood function that applies to count data. If it was proportions, you use a beta regression or whatever. You know, as I remember, the lasso regression is really built around, you know, real outputs. And just off the top of my head, wondering, you know, is that just how things are done?
I would love to talk to you more. I know that
Talk to him.
one of the reasons we set up a lab at Net is because, well, it hasn't been used for this specific purpose of predicting expression before. It has been used with epigenetic data before. So the most commonly known example is in the case of epigenetic clocks, which are basically models that to predict an organism's age based on its methylome. So how much DNA methylation it has, basically, and because animals get more methylation as they age, basically. And so this is the same type of methylation data we're using, whole-genome sequencing data, which is summarized with like basically a percentage, a methylation percentage, and lots of net regression is used to develop these methylation-based clocks.
I was imagining this as more of in a Bayesian context, so it's a lasso prior on the regression coefficient.
Well, I'm just thinking of the actual, if this was just a vanilla regression, putting count data in, depending on how small the counts are, that's not how we normally use.
But you could use, if you did it in the Bayesian context, your linear predictor could still.
Oh, your linear predictor, it's the response variable. Anyway. Again, if counts are large, it doesn't matter, but if they're small, sometimes the stats don't quite work out with. But then again, you're not using.
I would love to talk
P-values.
more because I've never actually taken a modeling class. It's all just like,
We can change them.
yeah.
Just past the good intro classes.
Yeah, so it's still based on like self-education. I would love to talk to the actual experts. Yeah.
Great presentation, Kathleen. Thank you. I think you hinted at it at the end of your presentation that you're going to try and compare this with physiological data. And so what that kind of sounds like is like, yeah, the current research is sort of based on like understanding coral responses, like the understanding of how like corals are studied is like around its sort of physiology and like its responses to its environment. And like what you're getting at is like this could like change the way we think about corals, where like if we can understand how genetically they're responding to their different environments, then like we could actually maybe understand what inherently like causes different like paths of like how like corals are suitable in one habitat versus when they aren't suitable in another. Because like I'm just thinking like yeah, if like you can understand like what genes are expressed in like what sort of environments, like that might give you like more markers than just like the biomass of like a coral in like the certain temperature regime. And like I'm curious, like this is super cool research, and I wonder like how do you think like if this could be used as like a biomarker, like how do you think this would change the way corals are studied?
I mean, this is another avenue in the world of epigenetics as biomarkers. Yeah. So there's lots of work in a lot of different systems using, you know, microRNAs as biomarkers of different kinds of stress. It's not necessarily the focus of my work, but it's definitely possible. Again, it would be limited by the low conservation of these features. So there are only 4 that are conserved among all the corals, and so if one happens to be, directly associated with whatever stress, then it'd be feasible as a biomarker. But if it's one of the lowly conserved ones, it might make things more tricky. Yeah.
I'm not seeing any questions online. Any final questions in the room?
Question about the like interacting depends like microRNAs, like you mentioned how all those layers are talking to each other. How would you is there a good way to model that instead of having all those like different interaction terms in the regression? I know you just for the sake of it, you get like thousands of them, you're not gonna do interactions across all thousands. That's crazy.
Yeah, I'm not sure. I haven't looked into it. Again, I'm like a biologist who's trying modeling rather than modeler who's working with biological system. So I am always open to talking with real modelers about how they approach things.
Thank you so much, Kathleen. That was wonderful.
School's out, enjoy summer break.
Great job getting through the quarter. Thanks for the